Hello everyone. Today we're going to discuss about the topic of DNA replication. It's very simple topic, not so difficult. So the DNA replication is a process in which a single parent DNA molecule becomes two identical daughter DNA molecules. Okay. So let's look at the structure first. So this twisted ladder structure is the DNA structure. Okay. As you all know that. And the railing of the ladder have two colors, as you can see. So these two colors symbolizes two different molecules which are the backbone of the DNA. Okay, and you can see the steps here, the steps these steps are the nitrogenous bases where the information has been stored, or we can say that the genetic information has been stored into these steps. Okay, so let's discuss about the railing first. So let's this is the one railing and this is the second railing, and the steps are the nitrogenous bases. Okay, let's draw the structure first. Let's draw the railing first. Okay, so the railing is made up of two components. The first one is phosphate group. Okay, and second one is the deoxyribose sugar, deoxyribose sugar. So the structure of the phosphate, it's a very simplified structure, or we can say that normally when it reacts with the deoxyribose, before it was a phosphoric acid. Okay, phosphoric acid, that's why we call it the DNA structure is acidic. Okay, so the structure is like that. So the phosphate group in the middle, so oxygen and double bond oxygen, and it has negative charge. But basically the phosphoric acid structure is like that: OH, 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 and double bond O. And when this phosphate group combines with the deoxyribose, with the phosphodiester bond, these hydrogen get released. Okay, at the end we get this kind of structure. Okay, let's discuss about the deoxyribose structure. So it has a pentagon structure. Okay, so let's draw the pentagon first. Oxygen in the middle, and this is the first carbon, second carbon, third carbon, fourth carbon, and the fifth carbon is outside. Okay, so fifth carbon is CH2OH. Third carbon is very important. It has the hydroxyl group attached to it, which is the OH group. First carbon is also very important, which connects the first carbon of the pentose sugar with the nitrogenous bases. Okay, so first carbon, third carbon has the hydroxyl group, and the fifth carbon is the connection point which makes a phosphodiester bond with the phosphate group. Okay, these are very important points. When we create the structure in the next slide, then we get to know about where the position of these molecules present, and these are very important. And the first carbon has the nitrogenous bases. Okay, so two things are clear in here. Okay, so first one is the phosphate and the deoxyribose, which are the railings, okay, so these railings, okay, these railings, okay, in the diagram, these railings. Let's discuss about the steps. Okay, so the steps are called as nitrogenous bases. Okay, nitrogenous bases. So what are the nitrogenous bases? Nitrogenous bases are classified into four parts. Okay, so the first one is nitrogenous bases. So first one is adenine, second one is guanine, guanine, Ag, third one is thiamine, T, fourth one is, anybody knows the fourth one? Cytosine, cytosine C. So AgTC. Okay, these are the four nitrogenous bases. And very important part, A always pair with the T and G always pair with the C. And the pairing in between them is double hydrogen bond. Okay, and G with the C has triple hydrogen bond. Okay, so you need to remember these two important points. Okay, so here you can see in the picture, in the image, A T, G with the C, T with the A. So okay, so A always pair with the T and G always pair with the C. So A and T has double hydrogen bond, G and C has a triple hydrogen bond. So here, so if this side is A, so that side should be T. If this side is G, that side should be C. If this side is C, this side should be G. If this side is T, this side should be A. And the bonding in between them is A and T is double hydrogen bond. G and C has triple hydrogen bond. G and C has a triple hydrogen bond. A and T has double hydrogen bond. So in the process of replication, so this bond, which is the hydrogen bond, gets breakdown, and we left behind single strand DNAs. Okay, so two single strands. So ladder gets broken. Okay, and we left with the on the left side we have the single strand DNA. On the right side we have the single strand DNA. Okay, I think you understand these three structures, which are the backbone of the DNA. Okay, so phosphate and deoxyribose acts as a backbone, which is the railing of the ladder, and nitrogenous bases is the one which stores the genetic information. Okay, let's get into the next slide and then we'll draw the ladder. Okay, and then you get to know the whole structure easily. Okay, okay guys, so let's draw the structure. Okay, so let's draw the pentose sugar first of the 3' side. So oxygen is here, first carbon has the hydrogen bond, second carbon hydrogen bond, third carbon has hydroxyl group, fourth carbon, and fifth carbon is outside the ring, CH2OH. Okay, now let's draw the next pentose and then we'll join the phosphate group with the pentose sugar. So this is the OH, one first carbon, second carbon, hydrogen bond. This is the OH group, third carbon, fourth carbon, and fifth carbon is outside the ring. Okay, so the bond formation is here. So the third carbon, which has the hydroxyl group, binds with the phosphate group, and the fifth carbon of the pentose bind with the phosphate group. This becomes the backbone. Okay, let's draw the next pentose sugar. First, second, third, fourth, fifth is here, CH2OH, and the hydroxyl group bind with the phosphate, and fifth carbon of the pentose bind with the phosphate. Okay, and last pentose. Okay, first, second, third, fourth, fifth is here, CH2OH, and this has the OH binds with the phosphate here, and this one binds to the phosphate, but it doesn't make any more bonding with the next pentose. This comes to the end of the chain, and where it connected with the fifth prime of the carbon. So this side is the fifth prime, and this side is the, because the hydroxyl group attaches to the third carbon, which is the start of the chain. So third prime, 5 prime. Okay, I think you understand the 3' and the 5' thing, as it mentioned in the image here. And the first carbon is this, first carbon is this, first carbon is this, first carbon is this. So imagine this is A, this is A always pair with the T. So this is G, G always pair with the C. So this is C, this is T. Okay, so we draw the strand. Okay, so 3' to the 5' side. Now we need to draw the anti-parallel strand, which must be, so if this side is 3, so this side must be 5'. If this side is 5', so this must be 3'. Okay, so let's draw this side also. So this is 3' left side, this is the 3' on the right side. So this one on the right side must be antiparallel, so we need to draw it opposite. Okay, let's draw it. Okay, so this is the pentose, First carbon, second carbon, third carbon, fourth carbon, fifth carbon is here, CH2OH. Okay, and then we'll draw the next pentose. Okay, so first carbon, sorry, this is the hydrogen or oxygen. First carbon, second carbon, third carbon, fourth carbon, fifth carbon is here, CH2OH. So the bond formation from here. Okay, so this is the OH group. So OH group makes a bond with the phosphate, and the fifth carbon bonds with the phosphate group. And this bonding is called as phosphodiester bond. Phosphodiester bond. Okay, so now let's draw the next pentose. First carbon, second carbon, third carbon, fourth carbon, fifth carbon, CH2OH, and this is the OH group makes a bond with the phosphate, and the fifth carbon makes a bond with the phosphate, and the last pentose. Okay, oxygen is here. First carbon, second carbon, third carbon, fourth, fifth is here, CH2OH, and this makes a bond with the phosphate, the phosphate, and CH2OH does make a bond with the phosphate, but this is the end of the chain. So this is the 5 prime side. Okay, so 5 prime, this is 3 prime, this is the 5 prime, this is the 3 prime. So these are the anti-parallel to each other, and then the first carbon has the nitrogen. So this side, left side is the A, so this side must be T. This side is G, so this side must be C. If this side is C, left side, and the right side must be G. On the left side, if it's thiamine, this one must be adenine. Okay, okay. So we draw the two railings, which are the backbone of the DNA strand. Okay, now let's join these nitrogenous bases together with the hydrogen bond. As you know that A always pair with the T, G always pair with the C, as I mentioned. The bonding between them is double and the triple hydrogen bond. So A and T has a double hydrogen bond, G and C has a triple hydrogen bond. Let's draw the bonding. So A and T has a double hydrogen bond, G and C has a triple hydrogen bond, C and G has a triple hydrogen bond, A and T has a double hydrogen bond. Okay, so this is the whole structure of the DNA. Now we are into the structure of DNA strands. Okay, now let's discuss about the process of the replication in this current slide. In the process of replication, this bond, this bond, double and the triple hydrogen bond gets broken, and we got a separated two different strands. Okay, now we need to add the complementary strands out of the template strands, and then we get the two different strands of the DNA at the end of the process of the replication. So let's discuss about the process of replication now. So the replication process has involved four enzymes. Okay, the first one is the helicase enzyme. Helicase enzyme. So this is the enzyme which breaks down this double and the triple hydrogen bond. Okay. Second enzyme is RNA primase. Okay. What RNA primase does, it gives the direction to the next enzyme, which is the DNA polymers, to start building up the DNA strand. Okay. So third one is DNA polymers. Okay, DNA polymers 1 and 3. And the fourth one is the ligase enzyme. Okay, so these are the four enzymes which are playing the game. If you remember these four enzymes, then you understand the replication process very easily. So the helicase is the zipper. It breaks down this hydrogen bond, okay, between the nitrogenous bases. RNA primase comes and start building up the the strand. But the problem with the RNA primase is that instead of thymine, it adds uracil, okay, which is incorrect in the DNA nucleotides. DNA polymers 1 and 3. So DNA polymers 3 is the one which build up the whole of the DNA strand. Okay, it follows the path of the RNA primase, which set up in the starting, and then RNA primase moves aside, and DNA polymers start building up the rest of the nucleotides. And the ligase is the one which joins any gaps between the pairing. Okay, we'll discuss in detail right now. Okay, so let's break down this hydrogen bond first, and then we add up the complementary DNA nucleotides. Okay, let me rub it off, and then we draw the structure here. So the bond has been removed, so the hydrogen bond has been removed. Okay, okay. So so helicase is the zipper. It comes and breaks down this the hydrogen bond between the base pairs. Okay, now second one, very important point. So RNA primase comes and start building up the strand. It build up couple of nucleotides, as it mentioned in the books. It build up couple of nucleotides, and then moves aside, and then DNA polymers 3, and it comes and then build up the rest of the DNA strand. But there is a mistake. This is a DNA nucleotide, and the DNA strand, we can say that, and it shouldn't have the uracil, as RNA primase adds the uracil. So let's draw the structure. So RNA primase comes and start building up the strand. Okay, so here is the so on the left it's 3 prime, so this must be 5 prime. Okay, so let's draw the structure. As I draw the structure before, let's start from here, the top one. Okay, let's start from the bottom one, easy to understand. Okay, so here is the structure, pentagon. Okay, 1 2 3 4, and fifth is here, CH2OH. Okay, and this is the work done by the RNA primase. So A bonds with the T, but RNA primase adds uracil instead of thiamine and makes double hydrogen bond. Okay, and here we got the fifth carbon here. So, and this is the phosphate group. So P is here. That's why it's 5 prime. This is the hydroxyl group to the phosphate, and then we got the next pentose sugar. Okay, and here first carbon, second carbon, third carbon, fourth carbon, and then the fifth carbon is here, bonds with the phosphate. Okay, and G bonds with the C. Okay, next let's get to the next one. Third one is here, so OH. Let's build up the next pentose sugar. The structure is a bit complicated, but if you keep on drawing with me, then you will understand the structure easily. Okay, so this one is the fifth carbon, CH2OH. CH2OH has the phosphate, and the OH combines here, and the C pair with the G pair with the C. So it comes G. Okay, and this is the OH here, and then we draw the next pentose sugar. 1 2 3 4 5, CH2OH, phosphate group attached to it. The last one is the hydroxyl group. This is the end of the chain. So 3 prime is here. Okay, and T with the A, because in the starting only in the start the RNA primers comes and add up the RNA primer, which is the U. But later on the polymers comes and build up the rest of the strand. So this is done by the DNA polymers, which is the third enzyme. Okay, and double hydrogen bond. So this work has been done by the DNA polymer. In the starting, only the uracil has been added. This is the mistake. We need to fix this mistake. Okay, this is the mistake. Okay, but rest of the strand is is done by the DNA polymers 3. Okay, so DNA polymers 3 builds up the rest of the strand. And you know that the and another very important point which I forgot to mention, which is the direction. So direction to build up the strand is 5 prime to the 3 prime side. So the direction here on the left side is 3 prime. So the antiparallel should be the 5 prime. So it's right direction. Okay. So the strand has been built up very easily. Okay. This is called as this strand of the DNA is called as leading strand because it's in the direction of 5 prime to the it's in the direction of the 5 prime to the 3 prime side. Okay, because here on the left side is the 3 prime, so the antiparallel is the 5 prime. So this is called the leading strand because the direction is fine. You know, it follows the rule, which is the 5 prime to the 3 prime side. Okay, now we need to build up the right side, which is the 5 prime side. This has some issues because the direction it breaks the rule, which is the 5 prime to the 3 prime, because it's already 5 prime this side. So, how the RNA primers build up the strand in the starting and the DNA polymers build up the rest of the strand. How it does it? Put the blue rectangle for you to better understand it, because we're going to start making the lagging strand, which is the most important part because it breaches the rule, which is the 5 prime to the 3 prime side. So let's draw it first. So we draw the pentose sugar. So oxygen is here, 1, 2, 3, 4, and 5 is outside here, CH2OH. So this one bond with the T. But here is the thing, because it breaches the rule, which is the 5 prime to the 3 prime. So the RNA primer builds the strand in the fragments, which is called as Okazaki fragment. So here is the one fragment, which is the pentose sugar and the fifth carbon. And here on the right side is the T. So here this side is the A. Okay, and it should be connected to the phosphate group. Okay, so here is the phosphate group. Okay, and we got the next pentose sugar. We got the next pentose sugar, 1, 2, 3, 4, and 5, CH2OH. But here is the trick. So the fragment is here to here. Okay, one fragment is done. So this one fragment is called as Okazaki fragment. Okazaki fragments, and there isn't any bond between this phosphate to the next pentose sugar. It keeps on growing like that. Okay, so so the bond has been broken. So this hydroxyl group of the pentose sugar doesn't make any bond with the, so this side is the empty. So this is the empty, doesn't have the bond in between them. Okay, so if this is C, this must be A with the T, G with the C. Okay, this is the phosphate, and there's a gap here. Okay, the gap here, this is the gap. And the next pentose we're gonna draw, next pentose draw, 1, 2, 3, 4, 5, CH2OH. This doesn't have, this pentose sugar doesn't make any bond with the bottom pentose. Okay, or the phosphate group. And then G with the C. Okay, we draw the next one, next pentose, which is the last one. 1, 2, 3, 4, 5. This is 5, CH2OH bond with the phosphate. This is the 5 prime end, and OH. It doesn't have the bond in here. This is the empty. So these are all called as the Okazaki fragments. Okay, these are all called as the Okazaki. So A with the T. So T here, double bond, G with the C, triple bond, and here is the G with the C, triple bond, and here with the double bond. Okay, so here is the mistake. Where is the mistake? Here is the mistake. U is here. Anywhere we can see the U. G with the C. So yeah, here. But but in this side, A with the T, Because RNA primers just start initiating the action by building up the strand, and it builds up a couple of nucleotides, and then the DNA polymers 3 comes and build up rest of the strand. Okay, so we end up, the lagging strand has the Okazaki fragments. So these are the Okazaki fragments. So these are the Okazaki fragments. So these are the Okazaki fragments because there's a gap between the hydroxyl group and the phosphate group. So here is the hydroxyl group and the phosphate group. This is the gap. Okay, so this is all done. Okay, but there is a mistake. Here is the U which needs to be replaced. So the polymers 1, DNA polymers 1 comes and replaces this U with the P and fixes up the mistake. So DNA polymers 1 is the quality checker. It checks the DNA strand whether it has been built properly, whether all the nucleotides are done properly, any mistake, it corrects the mistake. So here is a mistake which has done by the RNA primer. So it changes with the, it replaces with the P. Okay. So now we have the gaps here. Each fragment we have the gap between the hydroxyl group and the phosphate group. Here you can see, and the phosphate group and the hydroxyl group. Here is the gap. Here is the gap. Here is the gap. Okay, here is the gap. Okay, these are the gaps. Okay, because the DNA built up in the form of fragments. So these are the fragments. Okay, these are the fragments. Okay, and these are the fragments. These fragments are, as I already explained, these are called as Okazaki fragments. And lastly, we need to fix this one also, which is the gaps between the hydroxyl group of the pentose sugar with the phosphate group. So the last enzyme, which is the ligase enzyme, you can see here the fourth enzyme, the ligase one, it comes and seal the gaps. So ligase comes and seal the gaps. So it comes and seal and join the bond. Okay, it makes the bond. Okay, and this is the whole process of DNA replication. Okay, so the helicase come, zip it, RNA primase comes and build up the stand, build up couple of nucleotides, and it adds, instead of thymine it adds uracil, which is not the part of the DNA, which has been fixed by the DNA polymers 1. So DNA polymers 3 build up the rest of the nucleotides, rest of the strand, and the ligase joins the hydroxyl group with the pentose sugar, which is the joining point of the Okazaki fragments. I think this is the whole process of the DNA replication. I hope you have drawn the structure. I try my best to explain it because it's not an easy topic. It's a complicated topic. If you do it one more time and watch the video one or two time, pardon my mistakes. And yeah, that's the whole process of DNA replication. I hope you like this video. Thank you very much.